at the Food and Veterinary Department, and I'm the responsible for the National Reference Laboratory for viral contamination of shellfish. And what I'm going to present today is this cooperation, international cooperation project that was developed thanks to the FOODSEG, because the meeting with the Vietnamese part had started here. And so we are quite glad to present this project. We are in a quite an early stage because the project started last June, so our data are all preliminary. And unfortunately, our Vietnamese partners are not here today, so I will try to supply also the results of their research as much as I can. So why did we concentrate on seafood? Well, quite bluntly, because the European countries are large importer of seafood. Our main supplier is China, followed by India, Thailand, and Vietnam and Chile. Vietnam is more or less the third or the fifth, depending on which data you look to, supplier of the European countries regarding seafood. And beside that, Vietnam is also the main world supplier of this product that you all know, the Pangasius fillets. And Vietnam covers for more than 70% globally of the aquaculture supply. So it's a very large exporter of seafood products. Beside that, production is also for shrimps and prawns, so we also have crustacea and obviously clams and all categories of shellfish. Obviously, our interest is not only on the economic part, it's also on this food safety part. And I think that we can understand the importance of this kind of product by looking at the RAS alerts. As here I downloaded the RAS alerts of the last 5 years and the current alerts for 2015 regarding the seafood products, divided into categories, shellfish and fish products. And as you can see, despite the fact that fish products represent the largest part of the imports in the EU, the largest part of the alerts, information, border rejection regards shellfish products. And this is not really a news, because we have been known that shellfish is quite a risky product for quite a long time. And as you can see, besides the number, we also have a positive trend for the notification in the alert since 2013, when the ISO method for viral detection was developed. But as I said, the fact that shellfish is a risky product is not really new, as basically we have been known this since the Leviticus. So nothing really new, besides the fact that since the beginning we knew the problem, but we really didn't know why we had this kind of problem. In 1816, French physician Pasquier wrote a book on the danger of shellfish consumption. It was 40 years before Pasteur, so even if we didn't know anything about microbiology, we already know that there was some kind of hazard inside the shellfish. So it was more or less, I don't know why, but I'm quite suspicious of this product. What we also can see in the RAS alert is another point that relates to our project, the Vietnam part. As you can see, and I added the numbers in red, in most of the cases that were in which we had border rejection of products, the products involved came from Vietnam. So we have a problem with food safety, that is the presence on the market of products that are contaminated, but we also have a problem with commerce, because all the border rejection means quite a lot of money lost, both from the exporter and the importer. So why this problem? Well, this is because shellfish are filter-feeding animals, and they concentrate everything that is present in the water in which they go. A mussel, an healthy mussel, can filter up to 4 liters of seawater in 1 hour. So the ability to concentrate the pathogens is quite high. For viruses, for example, we know that the concentration inside the shellfish could be 2 log higher than in the environment surrounding the shellfish. So we can easily reach contamination levels that are dangerous. And what things do we find inside shellfish? Well, we have fecal bacteria, so Salmonella, E. coli, everything that can be discharged by sew pipe. Viruses, a very wide range of viruses, so we have noroviruses, hepatitis A, hepatitis E, adenoviruses, rotaviruses. But we also have autochthonous marine bacteria, so things that we do not discharge in the environment. They are the natural inhabitants of the marine environment, and they can also be human pathogens like Vibrios, Vibrio cholerae, Vibrio paramolyticus, or Vibrio sulnificus. And finally, we have algal toxins. That is also another danger that is associated with shellfish consumption. So, quite a large number of hazards to take care of. So here it comes the proposal for this project. This project was founded by the Italian Foreign Ministry and International Cooperation for the Italian part, and from the Vietnamese Ministry for Science and Technology on the Vietnamese part. And the main objectives were, first of all, methods of harmonization. In international trade, the use of harmonized method is of paramount importance for the reliability of the results, and moreover for the fact that in this way we can recognize the data and the results produced by the country involved in the exchange. The second objective of the project was method development and optimization, also through modification of the existing method. This because in food exchange, and especially for the exchange of a perishable food like shellfish, being able to analyze a large amount of sample in a very short time and with as much as possible load of work for the laboratories doing this kind of work is really important. So being able to simplify the method existing is quite important in this kind of trade. The last point was exposure assessment. What we are interested in is we don't really know the number, the levels, the quality, the types of pathogen present in shellfish imported from Vietnam, because no extensive analysis has been done, for example, on viral contamination in this shellfish. So after method of harmonization and development, our aim is to be able to perform monitoring program on the harvesting areas that are used for international trade. Just to be able to manage all the hazard, we try to put some kind of limits to the number of pathogens that we are looking for in this project. So we concentrated on enteric viruses, looking firstly at noroviruses and hepatitis E, because they are the most important in this moment in international trade, but keeping an eye also on other possible pathogens, Vibrio species, and specifically Vibrio paramolyticus, and finally this toxin, that is the polytoxin, that are toxins that are produced by Ostreopsis, that is a marine alga, for which there are still no limits on the products, but the limits are under discussion. So we try to put a step forward respect to the legislation. So how did we try to achieve our objective? For the first objective, method harmonization, 2 training sessions were organized in Hanoi, at the University of Hanoi. The first one was held during one of the FOODSEG meeting, the EU-Vietnam FOODSEG meeting, and so we could in that occasion present the ISO 15216, that is the reference method for viral analysis, and also present our electrochemical sensor for the detection of polytoxin that is being developed by the ISS, and the development is now almost finished with the test on naturally contaminated shellfish. So these were theoretical presentation, but we also had a chance to perform for 2 full days in the lab training session for a technician as well as student of the Hanoi University on viral detection. So it was quite useful to be able to handle the method for the partner. The training was completed with a second round in November. It was mostly dedicated to train on this method that has also been developed by ISS, that is a method for the enumeration of total and potentially pathogenic Vibrio paramolyticus. And these are the students of the Hanoi University. So this was for the objective 1, but to complete objective 1 we also had to, as I said, keep an eye on the viruses for which we still don't have reference methods. And as you can see from this data that reports the outbreaks associated with shellfish, the viral outbreaks associated with shellfish, there is not only norovirus and hepatitis A, there is also a problem with hepatitis E, astrovirus, rotaviruses, enteroviruses. So our question was which virus we should look for as our secondary objective after noroviruses and hepatitis A. So we performed a retrospective study on shellfish that had been sampled during 2008 and 2009 in northern Italy. This is the delta of the Po River, and we analyzed a total of 70 samples, including